and welcome everybody. Welcome to our NIHR Birmingham Biomedical Research Centre webinar. This is from our free webinar series, and tonight's talk is From Gut Bugs to Health, how changing the microbiome can help treat inflammatory bowel disease, often shortened to IBD. You may know it as that. And we are recording this so that we can share it with other people. But because you're on a live webinar, you'll be able to ask questions and we will try and get an answer from our guest speaker tonight. Our expert is Dr. Peter Rimmer. So Peter will be talking to you about this, and I'll just explain a couple of things first of all. But thank you very much for joining us today. We are delighted we've had so many sign-ups. We've had over 350 sign-ups for tonight's presentation. I know not everyone can make it on the evening, and say the great news is we record them as well, and you can watch back and have a look on our website, and you will find them all there. So we've done one so far, and this is our next monthly one. So I'm going to just do a couple of things to just explain really a little bit about who we are as well before we carry on to the talk. And wanted to just let you know that we are funded by the National Institute for Health and Care Research, which is the funding arm of the NHS, if you like, or it's that centre. And what exactly do we do? Well, we do a number of different things, because we are actually a collaboration between the university and the trust in each case, because there are actually 20, including us, of these biomedical research centres throughout England that are funded to do this work as collaborations. So although we might be based in Birmingham, as you can see with a number of different partners that we work with, we are actually much wider than just Birmingham, and the research that we do is something which is, worldwide. It has effects across the whole world, and as I mentioned, we are publicly funded, and what we're trying to do is make sure that we turn the scientific discoveries into diagnostics and better treatments for people. And our research centre is really focused on inflammation and making sure that we improve the lot of people with conditions that are affected by inflammation, which can be a healthy part of recovery, but sometimes inflammation can go too far, and that's why it's really important for us to understand more about it. But I'm not the expert. I'm going to be passing over to you in a minute, Peter, who is the expert. And if you do want to ask him questions, what you can do is, if you have a look on your toolbar, you'll see there's a Q&A icon, which you can click on, and we'll be selecting them. And if we get lots of like similar questions, for instance, we'll pick, a question that identifies all of that rather than ask every single one. We can't give any medical advice. That's not the purpose of today's call, but we do wish that you would please ask questions. We're delighted to hear from you, and I will talk to you after the presentation when I'm hosting the Q&A. So thank you very much for joining tonight. So at the moment we've got a number of different people on the call, and we're really pleased to see you. So without further ado, over to you, Dr. Peter Rimmer. Thank you.
Well, thank you very much for the introduction, and thank you for giving me the opportunity to talk today, because I think this is something I spend a lot of time working on and a lot of time thinking about. So to get the opportunity to come and talk about it, and to talk about it to members of the public as well, is something that I think is a really fantastic opportunity. So as we discussed, the title, From Gut Bugs to Health, How Changing the Microbiome Can Help Treat Inflammatory Bowel Disease. Now, as you know, my name is Dr. Peter Rimmer, so I'm a consultant gastroenterologist at the Queen Elizabeth Hospital in Birmingham, and I'm a researcher within the University of Birmingham, working as part of this biomedical research centre. So if I get this, get slides moving, there we go. So what are we researching? So my theme is oral and systemic health, and I'm working with other researchers as part of that theme to really try and understand how the microbiome contributes to the onset of inflammatory bowel disease and how we can look at those changes in the ways in which it contributes at that early stage of the disease to try to help the way we treat patients to get the disease controlled more quickly and to keep it controlled in the longer term. To facilitate that, we've set up a rapid access clinic for patients who are suspected of having inflammatory bowel disease. So we've taken the funding that we get from the research that we do, and we've been able to use that to try and get patients into our clinics more quickly, get them a diagnosis at an earlier stage in the duration of their symptoms, and get them on the right treatment sooner. That involves using the standard treatments, but what we'd like to do moving forwards is to try and conduct a study using fecal microbiota transplantation, or FMT, which I'll speak a lot more about as we go through this presentation, in newly diagnosed inflammatory bowel disease. And at this stage, that's specifically focusing on ulcerative colitis because that's where we have the most data and research done already, and that's where we have the best signal that suggests it might be effective. And we hope that by intervening sooner, we might be able to boost the responses to our traditional treatments, but also get the disease controlled using the FMT itself, and then keep it there longer by getting those sort of chronic inflammatory pathways controlled at an earlier stage. There are some core concepts that you need to understand for me to talk about the microbiome and how we might try to change it. And so we'll spend a little bit of time going through those, and then we'll focus in on the treatments and how we might be able to do things differently. So what is a microbiome? So I think this is important because it's something that's discussed very broadly now in multiple health conditions, but also as almost like a lifestyle measure as well. So a microbiome is something that is present in all different environments. It's not something that's unique to us in humans. So if we look in the soil, we'll see a unique microbiome. If we look in the ocean, we'll see a unique microbiome. And across humans, we have multiple different unique microbiomes. So you'll find that in our mouths, you'll find it in our digestive systems, you'll also find it on our skin. And for example, the vagina has its own defined microbiome as well. And there are core terms that we'll use when we're talking about it. So microbiota is something that's often confused with the overall microbiome, and that refers to the individual organisms that make up the overall community within a given environment. And that's bacteria, but it's not just bacteria. It includes viruses, it includes fungi, it includes archaea. So it's important that it's not just bacteria and it's not specific to us as humans. And then when we talk about all of those microorganisms together, including all of their genetic material and what they functionally contribute within a specific environment, that's when we're talking about the microbiome. And I think it's really important to emphasize that the kind of, we use the word symbiotic, but it's the positive relationships, the way in which we help each other. So the way in which the bacteria can benefit us and other animals is really fundamental to the way life is as we see it today. And those relationships have been there for millions of years, developing in the background. So what does the gut microbiome do for us? Well, it helps us to break down nutrients, so to break down indigestible carbohydrates and to produce what we call bile acids, so secondary bile acids in particular, which are something our body's able to use. So the cells within our bowel lining are able to use these bile acids to help stay healthy in essence, but also to help maintain this barrier. So there's a mucus layer that sits on our bowel lining that helps form a barrier between the immune system and that's housed within the bowel lining and the contents of what's inside of our bowel, so the poo. And if there's a breakdown in that bowel lining, it can lead to immune responses that can be excessive and dysregulated at times. So it's important that our cells are able to maintain that, and it's important also for them to use these things that are broken down by our microbiome to stay healthy. It helps control the way that our immune system works and helps our immune system to function. So it helps us produce proteins that can help combat bacteria and other microorganisms. It regulates the production of some of the inflammatory messengers, so some of the things that our cells produce and able to communicate with each other. So if there's a particular type of blood cell in an area that's identified a bug and wants more cells to come out, they might use these cytokines or messengers to communicate. And it supports the function of our immune cells as well. It also helps us to break down different medications that we might be given so that our bodies are able to use them. So how do we measure the microbiome? So in the past, we would try to grow the bug. So we use this process called culturing, where you'd put a sample in a specific media, and by media we mean something for it to use to grow, almost like food, and we would see what bugs would grow in that. But there are lots of problems with that approach. So it takes a lot of time. It's very labor intensive. And not all bacteria, for example, and certainly not viruses, but not all bacteria can be grown. It's not necessarily going to be possible. And the media or the thing that you use to help it to grow might not be the right media for all different bugs. So what you're doing will inherently be biased towards particular organisms. And that means that you might be able to identify some bugs, and you might be able to identify that they're alive and they are doing something because they're growing, but you can't get an overall idea of all the different things that are forming part of that community. So if we go back over the last 20 years, we've seen the development of this kind of set of tools called next generation sequencing. So the first one we talk about is this 16S RNA sequencing. So it's a complex term, but we take some of the material that's within the ribosome, so a particular component of the bacterial cell, and within that you will find some genes that code for it, and within those genes you will find some parts of it which are common across all different organisms, and some parts of it that are unique to that specific organism. That bit of coding is only found in that organism. So if you can amplify that up so that you can read it at scale, you can then identify just from that bit of code what organism it is. And this can be done at quite low cost now, and it allows you to get information about what bacteria are there. It doesn't tell you whether those bacteria are alive or dead. It doesn't tell you what they're doing to contribute to a microbiome. It just tells us that they're there. Shotgun metagenomics is a higher cost approach that takes all of the genetic material in a given sample and sequences it. So if we do that with our poo samples, for example, you'll find lots of human DNA in there, but you also get all of the microbial DNA, and you can try to do some clever things once you've got that DNA and you've sequenced it to figure out what the bacteria are, both deeper down to identifying individual species, but you can also tell to an extent what those bacteria are doing and how active they are from a metabolic standpoint. Now, that doesn't tell us what they're producing. It just is kind of you're estimating from the activity what they might be doing. So there's another way that we can measure what the microbiome is doing, and that focuses on metabolomics. So again, another complex term, but it is just measuring some of those short-chain fatty acids, some of those bile acids that are being produced by the bacteria that are in our bowels, things like butyrate and acetate that you can measure directly to see that in the context of, for example, if there's more of this specific type of bacteria, do we see more of these end products being produced as well? And things that we would know to have more of would be considered healthy. So if we focus specifically on bacteria, it's important to know a little bit about how they're classified, and we'll use humans as an example. So this is what's called a taxonomy, which is just a fancy term for classification, really. And so if we look at humans, you would come down to find Homo sapiens, which is, it is humans. But if we were to talk at a family level, which is what 16S does to an extent, you wouldn't be able to separate out humans from gorillas. It would broadly group us together. Now, 16S sequencing does allow us to get beyond just family to genus, but that wouldn't be able to separate humans from Neanderthals, for example. Whereas if we were to do metagenomic sequencing, then it allows us to get down to that species and at sometimes even strain level. When we look at the human gut microbiome or microbiota that can constitute the microbiome, we look at a phylum level and you'll see that 65% of our microbiome is constituted by these firmicutes and 30% by bacteroidetes, with a small proportion of a series of other different phyla. However, that is a very broad classification. So when people use the term firmicutes, that's the same as classifying things broadly as a group amongst humans, well, not even humans, mammals, birds, fish, reptiles. So whilst you'll see lots of different terms used, it's important to know at what level they're talking to be able to separate out, if you're talking about a phylum of bacteria, there'll be multiple different things within that are all doing different things and are all contributing in different ways. So it's important to be able to look at a much finer level to be able to tell what all this means. So what does good mean when it comes to the microbiome? And this is a really challenging question, both for us in the field, but also for people who might be paying for commercial microbiome testing, which is available now, and they'll come out with some measures, and it's about understanding, okay, what does this mean for me and how have they decided that? So you see there are different companies now. So I use Zoe as an example here, and they'll comment on the relative abundance that you have if you submit a sample of good and bad bacteria, and they might give you a score, like something like richness, or they might give you a gut score. Is this good or bad? And they'll tell you, well, you might have this problem because you've got some of these bacteria. And how have they decided that that is a problem? And how are they defining what should be healthy? And that is one of the core concepts in understanding the microbiome is what is healthy? Because there are lots of different things within our environments that are contributing to how our microbiome is put together. And so healthy is something that people have spent a lot of time debating. Is healthy just the microbiome in the absence of a defined disease, or is it the absence of bowel symptoms? Many people will have bowel symptoms from time to time without having an organic disease. So how do we define it in that context? And how do we define it across different geographic areas? So a lot of work, and there was something called the Human Microbiome Project in North America, which tried at great length to quantify what the microbiome was in healthy North American people. But can you apply what's defined as healthy within those patients, or those people, because they're not patients, to patients or people who are in less urban environments, less economically developed areas that might have fundamentally different diets and fundamentally different exposures from day to day, all of which will affect the microbiome. And within the published data set, there is a broad underrepresentation. So there's not as many people from less economically developed countries that are participating in this research. And so we don't know if what's healthy in North America is healthy everywhere. Once you've settled on what definition of healthy you're going to use, then you have the specific measures. And a lot of these scores that these different companies come up with will be based upon alpha diversity. So that is a measure of the number of different bacteria that are present in a sample. And when we use richness, which is a form of or a component of alpha diversity, it's not just the number of different species that are present, but it's how much of each of those species is present as well. And that usually generates a specific value. So you can say that you have got a set score for somebody's microbiome in that particular sitting. And that is helpful to us because you can compare those set values amongst different patients or different groups. Beta diversity is another measure that we use, and it's a specific comparison between two different populations. So how similar two pairs of samples are. That might be between two different people, or it might be between groups of people who have, for example, an inflammatory bowel problem and another group of patients or people who are healthy. And so you will normally have a score that comes from that, but it's often represented as a plot that shows the separation, the distance between the two different groups and how similar they are. That doesn't tell you about specific bacteria. This is just a plot highlighting what we mean when we say alpha and beta diversity. But when we want to try and understand about specific bacteria, then we'll talk about this thing called differential abundance analysis. And that allows us to compare the numbers of individual bacteria that are present across different samples or different groups of samples, whether that's disease groups or individuals. So what about inflammatory bowel disease? Obviously, I've spent a lot of time talking about the microbiome there. So what do we know about inflammatory bowel disease? Well, inflammatory bowel disease is broadly split into ulcerative colitis and Crohn's disease. They are chronic conditions that are characterized by periods where the disease is really active and flared up and people are having lots of symptoms, and periods where it can be quieter, more controlled. Ulcerative colitis is limited to the large intestine, and it causes inflammation that's more continuous in where it's involved, whereas Crohn's disease can cause areas of deeper ulceration and inflammation within the bowel, but there might be patchy areas of normal bowel next to it. Crohn's disease can affect any part of our digestive systems, and that includes from our mouth all the way to changes around the back passages, south, what we call perianal Crohn's disease. Both of these, when active, can be really life-altering, disabling conditions. So patients will experience severe diarrhea and bleeding. There can often be this real urgency to go to the toilet such that it's not possible to make it to the toilet in time. It can cause significant pain. It can mean that people are profoundly fatigued. And many people will describe having to plan their lives around where the next toilet is going to be. And some people feel unable to leave the house due to the severity of their symptoms. So we do recognize it can be a hugely impactful condition. The cause of inflammatory bowel disease, which my slides have just decided to stop moving. There we go, is a combination of different factors together. So we have our microbiome that I've spoken about, which is influenced by lots of different environmental factors, which can often be the things that might trigger this off. An impairment in that barrier function of that mucus layer I've spoken about, but also genetic factors that mean once the immune systems become activated, it then isn't brought properly under control. And that immune response is allowed to continue unchecked and cause all of these problems and symptoms that patients experience. So am I, my slides moving again? There we go. So when it comes to understanding the microbiome in inflammatory bowel disease, there's been quite a bit of research done, but really there is a bit of a phrase, but the devil is in the detail. So we've done a bit of research looking at all of the published studies that talk about the microbiome in patients with inflammatory bowel disease that haven't had any treatment yet. So we can say that it's the closest to the onset of the condition possible. And we've looked at the studies that have presented one of these measures of diversity, this alpha diversity in patients with inflammatory bowel disease. And we've looked at comparisons with healthy people, and we've looked with comparisons with people with symptoms. But there's so many different methods that are used to generate these values that it's really hard to compare. So if we look at just comparing the different types of sequencing, the different types of methods that have been used to get the DNA that we've used the symptoms to the sequence, and the different sample types, you can get over 13,000 different combinations of methodology before you even talked about the different sort of bits of technology they've used to actually analyze the data once you've generated it. So really, you're not comparing like for like, and it can be very challenging to interpret. So what have we been doing in Birmingham? So we have established this rapid access clinic that I've mentioned, and that clinic is combined with research to allow us to see patients who are suspected of having inflammatory bowel disease more quickly. And in doing so, we've seen over 1,200 patients now encompassing the 4 sites of University Hospitals Birmingham. So that's Queen Elizabeth, Heartlands, Good Hope and Solihull. And we've reduced time from initial referral to being seen by half, certainly within the Queen Elizabeth Hospital. And we've managed to get 300 patients who went on to be diagnosed with inflammatory bowel disease involved in our research, which has allowed us to understand a little bit better about what is going on with the bacteria in their bowel, specifically looking at poo samples. We've also been able to look at blood samples and whether there are any things in the blood that allow us to predict how a patient might do with treatment and how we should be treating them earlier on. And we've also been able to look at some of the cell populations that are present in the bowel lining, so we can understand how the bacteria that's within our poo, how that's interacting with those inflammatory cells that help drive the condition. And what we've done is we've taken our own microbiome data and we've linked that with the microbiome data across all of these studies that I mentioned that talk about diversity data. And we ended up identifying 18 different studies across different continents where they'd presented microbiome data. And we tried to put our data with that together so that we could put it through a single point of analysis and then try to adjust for all of those method problems that make it hard to compare, so that we could get a core picture of what are the changes in people who present with inflammatory bowel disease. We see that broadly there is a reduction in the alpha diversity, particularly amongst adults who present with inflammatory bowel disease compared to healthy people. But we don't see that change so much when you compare with people who have bowel symptoms but not inflammation. So we see a lot of people who might have irritable bowel syndrome, which can also be quite disabling. And we see that those patients also have significant changes in their microbiome. So that tells us that the microbiome is important, but it's not the only piece of the puzzle. And it's not the only thing that's on its own is driving this process. So when we compare patients with inflammatory bowel disease, both with those patients who have bowel symptoms but not inflammation, and those people who are healthy, we see an increase in a lot of these bacteria that are typical of the mouth. So there are lots of bacteria that are present in our mouths, and actually our mouths generally have a lower diversity in health. But when these bacteria make it into our digestive systems, which is not unusual, but in the right environments and in an inflammatory environment, we see that there's more of them. And there's these bugs like Fusobacterium nucleatum, which also has been linked with colorectal cancer as well, that are seen in greater numbers. And it's not only that they're seen in greater numbers in people that have inflammatory bowel disease, but they're also seen in greater numbers in those patients with inflammatory bowel disease that have more severe inflammation, and in those who go on to need our stronger treatments earlier on, because the first medications that we use don't control things as well. So it's not only associated with the disease, but it's also associated with having more difficult to treat disease. So if we know that there are these changes in the microbiome and there are these changes with oral type bacteria, how can we try to change it? So there are different ways that we might approach that. So we talk about using additive approaches where we add something to the picture, and that's what I'm going to talk about most, which is fecal microbiota transplantation. There's also live biotherapeutics, which I'll talk a little bit about what that means, but really the focus is going to be on fecal microbiota transplantation. I'm struggling to get this. There we go. You can try taking something away. So, for example, using an antibiotic to target a particular group of bacteria that you want to remove from that individual's microbiome. And you can try more gradual modulatory changes. So things like using probiotics, postbiotics, prebiotics, or making changes in somebody's diet to make a gradual change. But as I say, I really want to shine a bit of a spotlight on fecal microbiota transplantation. And this is a little kind of frozen fecal microbiota transplant that we might then go on to give to a patient. So what is fecal microbiota transplantation? Well, using this type of approach to a health problem is something that has been spoken about for a long time. And so if you look back through records, you'll first see it mentioned in traditional Chinese medicine from the 4th century. Obviously, things have been refined a little bit since then, and the mainstay of its treatment has been in conditions such as Clostridioides difficile, which is a type of infection associated with antibiotic use. And in patients who are then given antibiotics for this Clostridioides difficile infection, we can see that an FMT is able to cure 90% of people who haven't responded to those antibiotics. So how do we get an FMT? Well, you have a donor stool sample, which is very carefully screened, both in terms of looking in the poo for particular bacteria or other pathogens, looking in the blood to make sure there aren't any viral infections, and very carefully health screening individuals. You then will mix it with some saline, which is just salty water, and usually some glycerol as well. And you will filter it, but not filter it to such an extent that all the bacteria is gone. You want all the bacteria to still be in there, but there's other things in there as well, like short chain fatty acids that we mentioned, like that butyrate. And you then administer it. And there are different ways of administering it. So for Clostridioides difficile with a C. diff infection, we use a tube that goes via the nose into the stomach, and you give a single dose via that tube. And that is able to bring about that 90% cure rate I mentioned. But in chronic inflammatory conditions such as inflammatory bowel disease, usually you need repeated treatments, and it's necessary to use to deliver the treatment in a different way. So we find that the most effective treatments are those when they're given directly into the large intestine, and we would usually give the first dose via something called a colonoscopy, and then subsequent doses are given just as an enema. And so that's just as a infusion or an injection just directly into the bottom. So this is the University of Birmingham Microbiome Treatment Centre. So this is a big part of the research that we do and the service that we provide. So it's a lab that's licensed by the MHRA, which is kind of the central agency that monitors how drugs and other medicinal products are produced in the UK. And it's been running since 2017, initially as part of a trial, but now supplying FMT to treat C. diff across the United Kingdom. And then it's providing the material for the trials that we hope are currently conducting and hope to conduct in future. So the first trial that we conducted was this study called Stop Colitis. So we took patients with ulcerative colitis who had persistent active disease. We intervened with fecal microbiota transplantation. So the first dose given a colonoscopy and then subsequently given via 8 enemas, or 8 weeks total treatment. And we were able to see that in patients who responded, which a number of them did very well, so 75% of patients who received it via colonic delivery responded. The study was based upon proving that this mode of delivery was more effective than nasogastric tubes. And certainly this study demonstrated that significantly, with only 25% of people who received it via an NG tube responding to that treatment. And in those patients that had the colonic delivery, so it was given as colonoscopy and enemas, we saw a number of different changes. So the number of different bacteria that they had in their fecal samples increased, or in their poo samples increased. We saw more of this particular group of bacteria called Clostridia. And we saw increases in the numbers of these short-chain fatty acids I mentioned, like butyrate, in the poo samples from those patients. We saw that those changes in the bacteria that were present were associated with changes in the the white blood cell populations that were present in those samples. So more of the white blood cells that are involved in regulating our immune responses and less of the white blood cells that are involved in driving ongoing inflammation. So what have other studies said? Well, there are now 9 studies in total that have compared FMT, so fecal microbiota transplantation, to a placebo in ulcerative colitis. And by placebo, we mean something like, a sugar pill. In this case, it might be saline, or it might be giving the patient their own poo back as a transplant. So it's something that the patient doesn't know whether the medication they're receiving is the thing that we think is going to be effective, so in this case the FMT, or something that we think wouldn't be effective, so just a sugar pill, or giving the patient their own stool back. And those studies, when you look at them all together, they do present a clear signal that FMT can be an effective treatment for patients with ulcerative colitis. But there are problems as things stand at the moment. So it's really labour intensive to do. You've got that careful donor screening. You've got to then produce the FMT, and then you have to bring the patient back to hospital. In our study, we did 8 weeks, although we actually saw that most patients had responded by their 4th treatment. So even 4 weeks of weekly visits to hospital whilst you go through the treatment. And that presents a problem with scalability. So within the NHS at the moment, the resources are strained. So can you justify doing that at scale? We're doing it within a defined study. But if you had to do it for everybody with ulcerative colitis, clearly that would be a lot more, and it would be hard for us to deliver that. Studies have looked at making FMT capsules, and that is one way that can help combat that issue of coming back to hospital repeatedly, but it doesn't necessarily get over the challenges you have in getting donors through screening. Only 1 in 10 make it through, and then being able to produce those on a large scale. There's been interest in using these novel biotherapeutics. So what people have done is taken studies where we've done FMT, looked at the bacteria that have gone up in people who have done well, and just taken those bacteria alone and put those in a capsule that you can then produce at a much greater scale. But we have no idea whether those will be as effective as FMT, because you're taking away a lot of the other things like the short chain fatty acids that we think are really useful. We still don't know what makes the best donors, and we know what makes safe donors, but do we know what makes the donor who's going to be the most effective at treating bowel inflammation? There's been lots of work looking at this, and lots of studies have sort of tried to speculate and try and demonstrate that their donors might be better for a particular reason, but we've yet to see a consistent signal from that. We know that people who have long-standing inflammation and who have severe inflammation are less likely to benefit from FMT. So we know that it's not going to be effective for everybody. And I get that to move on again. And we do see that in people, even when they respond to the FMT, so if we give them the treatment, the disease is completely controlled, but we do see that the inflammation can recur when the treatment stops. So there was one study where they used pooled FMT from multiple different people. And for those patients that they got into remission, they said, right, how are things after a year following on from that treatment? And over half, so 58%, had found that their disease had flared up once they stopped the treatment within that year. Another study used capsules, and they let 4 people carry on taking the capsules after they'd done the study, while 6 of them had to stop. And these were all people who'd got completely controlled by the treatment. And all of them that carried on taking it were able to stay in remission, whilst all the people that had stopped had had a flare up by a year. And actually, most of them, it was within half a year that things had flared up. So what do we plan to do next? So we want to do more to understand how these oral bacteria that make it into our bowels, how they behave, how they get there. And so we're doing much deeper characterization of what bacteria are present in the mouth by looking at saliva, but also by looking at dental plaque. And we're trying to do a much deeper level of sequencing than what we've done before to try to understand that at a sort of the most fine level, I suppose. We're trying to look at saliva to see whether there are any ways in which we can differentiate out people who are referred to us as having an inflammatory bowel disease or not, using less invasive approaches. And that's something that's in its early stage, but has promise. We're hoping to undertake interventional studies using FMT, and we've got a grant in, which I'll talk about in a little bit more detail, which is called HALT colitis, and that's specifically targeting people who have just been diagnosed with ulcerative colitis to see if we can get their disease more controlled and see if we can get more people in remission after 12 months. And we're also doing a study in PSC IBD. So PSC is primary sclerosing cholangitis. It's a condition and another inflammatory condition that affects the bile ducts of the liver that at the moment we don't have any treatment for, and it can be progressive, and we'll find that some people need liver transplantation at a young age, and that's strongly linked to inflammatory bowel disease, with the majority of patients with PSC either having inflammatory bowel disease or developing it at some point in their life. And we want to study in more detail the way that FMT, so fecal microbiota transplants, how they change as we process them. So what gets donated as it goes through that processing to be then finally administered to the patient? Are there changes as it goes through that might make it less effective and that might allow us to kind of better look after it, really, to have the best effect for our patients? So this mentions Holt colitis, which we're hoping to get additional funding to undertake in our ulcerative colitis patients. And then we have additional funding from Bowel Research UK to look at the composition of FMT, or what is in it as it goes through all those different stages. So that's kind of our focus and how we hope to move forwards. And there's a lot of people that I need to acknowledge as part of this. So obviously the NIHR Birmingham BRC patient and public involvement and communications team have done a lot of work helping me to put this together and hopefully do it in a way that's been useful. And there's been obviously the funding that we get from both the BRC and additional funders like charities like Guts UK. The PhD students that I work with, so Virelia Stoiker and Ekin Ye, and collaborators within the NHS Trust, so the support of our theme leads, so the people that are really overseeing what we do, Tariq Iqbal and Ian Chappell, and all of the clinicians that I work with, all of the other gastroenterologists and specialists in oral health, and all of the collaborators within the university that do the fantastic work and facilitate all the science that we do to try and understand and improve outcomes for our patients. And of course, all of the patients who have participated in the work that we've done to allow us to try and improve things and to allow us to try and understand more, because certainly a lot of these benefits that we hope to gain are in the long term. So we do appreciate that patients, when they do participate in our studies, are doing it for that long-term benefit of not just themselves, but the other patients that we're treating. So now moving on to the questions and answers.
Right, so back to the question and answer session. Sorry, I went the wrong way on the slide. So Peter, we have had some questions already, which is super, as I said. And the first one that I'm going to ask is, and again, forgive my pronunciation, how do more oral pathobionts end up in our stools?
Yeah, so it's an important question, and I think there's a lot of debate around this, I suppose. So we know that obviously our mouths and our digestive systems are one continuous tract. So these are things that we're swallowing, but there are also things that enter our bloodstream as well. And there's this link with inflammation in the mouth, so what we call periodontitis, and inflammatory conditions. So it's not just what we're swallowing, but also what's entering our bloodstream. And one of the areas of big debate is in inflammatory bowel disease, the reason that you're seeing more of these bacteria simply because all the other bacteria are being wiped out in an inflammatory state, and that these ones are still continuing to get there because we keep swallowing them. I think there's more evidence that would suggest that it's not just that, and that these bugs do seem to have a role in contributing to ongoing inflammation as well.
Brilliant. Okay, thank you very much. The next question, would FMT be useful for Crohn's disease in patients with ileostomies?
Yes.
So people without a large bowel and just a small bowel.
So this is, again, this is a really important question for us to try to answer. And I kind of glossed over a little bit. I was going to spend some time, but then I sort of condensed it right down about other types of inflammatory bowel disease. So in Crohn's disease, there are some studies that look at it, and have also been looked at in patients who are post operations, although it's been looked at mostly in people who have had a join made between the bowel rather than having an ileostomy. And we don't have clear signals for it being effective at the moment. So there are less studies, there are inconsistent results. So it's not just one pattern where it definitely says it's effective. There's a big Australian study going on at the moment, which is the first proper kind of placebo-controlled one, which I kind of explained a little bit earlier, which we're really interested to see the results of. There was a study from China published just this year that looked at using it alongside making some changes in diet that seemed to suggest it might have some effect, but there were lots of problems with the way that study was conducted. So I think the answer is we don't know for sure. It hasn't been ruled out as being effective in Crohn's disease at all, but I think we certainly feel like we need a bit more evidence at the moment to be sure, and it's not as clear as it is in ulcerative colitis. And then, yes, within the particular type of Crohn's disease as well is another question. And certainly patients who have an ileostomy and have found that the inflammation has come back in their small intestine are usually a group of patients who have had more difficult to treat disease over the years. So it would be a really important question for us to answer, but not one we can be certain of at the moment.
Okay, well, thank you. Next question. Would FMT also be potentially used to treat food allergies?
Well, I think there's a really broad interest in different applications for FMT in treating all sorts of different things. And I think, do I have the knowledge to say that FMT could be used to treat food allergies? I don't know, is the honest answer. It depends on whether it's kind of intolerance, whether it's true allergy. There's lots of different factors that would contribute to that. I think at the moment we're away from that, but I think this is something that's going to grow and grow. So we're really in the early stages of applications for this. So we have, established C. diff treatment. It's there. We know it works. In IBD, we're kind of exploring. There's lots of questions about whether you could use it for intolerances, whether you could use it for this kind of broad group of functional bowel problems. So as I discussed in patients with IBS, we see that there are defined microbiome changes in those patients. It's not all people just sort of imagining symptoms that they don't have. It's a very real thing. So could we then start intervening in those patients as well? And these are all questions that will come in future. But certainly at the moment, I don't have the kind of evidence that tells me whether they'll be effective or not.
Okay, thank you. And can I just remind people, we can't do this any other way than using this Q&A button. So at the bottom of the screen, you should see a box which has got a question mark in that says Q&A, or an icon that looks like that, because we can't, people can't talk. So there's no possibility if you've got your hand up. And I think that has happened for someone. So please do keep using that. It seems to be working really well, because we've got some other questions, Peter. So I'm just going to keep firing this at you as long as we've got, if that's okay, and until
Yeah, that's
7
fine.
o'clock. Thank you. Right. So, and again, interestingly, there was a question kind of related to diet again. This one is about, does a plant-based diet help the microbiome, do you think?
Yeah, so I think that again, a lot of these companies will talk about different diets when they're giving you the commercial testing as well. And I think there are the things that we've got pretty robust evidence to say is that certainly a diet that's lower in ultra processed food, that's lower in highly refined sugars, is going to be one that will be better for your microbiome than a diet that contains lots of those. So a plant-based diet would fall within that. Certainly. Can you say that it would be better than any other diet that excluded those or minimized the amount of those things that you're having? Difficult to be so certain of, but we know that diet can impact on the microbiome. And one of the FMT studies that's within the literature used this kind of anti-inflammatory diet alongside FMT to see if they could make it more effective. So I think changes in diet is something that we would want to see alongside an FMT intervention. And I think when we do these studies, we very carefully look at what patients are doing with their diet. And I think part of that is because when you've got people with really bad bowel inflammation, their diet will become very, very restrictive, understandably, just to try and make things that little bit more manageable. So then if we get the symptoms a bit better, we'll find people might then start bringing some of those things that they'd restricted back in. And so that can impact upon your microbiome as well. So certainly, I would be happy to go on record as saying a diet that's low in ultra processed food and refined sugar, of which a plant-based diet would be one, would generally be positive for your microbiome relative to one that had more of those things.
Thank you. Next question is, would oral therapy be part of the treatment for IBD in the future, given the influence of the oral microbiome on it?
Yes, and so that's something that is even more kind of, I suppose, difficult in some ways. So there's a lot of interest in the oral microbiome, and I've been at talks today that have focused on that and ways in which you can improve the oral microbiome. And so I think it is something that is being looked at. And there's lots of interest in developing this concept of oral microbiota transplantation. Could you do the same sorts of things as we're doing in the gut into the mouth? And that's something that we're working on as part of this BRC theme, looking at ways in which you can encapsulate bacteria and get them to engraft in the mouth, which obviously is an environment that's exposed to more extremes in the way in which you have a cigarette, you have a coffee, whatever it might be, that's going to have a direct impact on your mouth. So I think it has to form part of it. There's components of it that are hard to understand. So obviously we talk about periodontitis, this kind of gum inflammation being associated with inflammatory conditions, yet many of the patients who develop IBD are very young and don't have periodontal disease necessarily. So there is more that we need to understand, but that is a big part of what we're trying to do with this. And with some of the technology that's available, you can kind of almost track these bacteria as they start in the mouth, whether it's and see whether they're making it to the large intestine. When you see these bacteria increased, can you demonstrate where they've come from? And that's all part of what we're trying to do.
Okay, and perhaps slightly connected to that, um, do you think probiotics make a significant difference in the health of the microbiome?
Yeah, so I think there's a lot of work going on at the minute, looking at different types of pre and postbiotics and looking at all these different things that might have an impact. And so the probiotics that are kind of broadly available at the moment don't have a great evidence base for IBD, but that's not to say that there isn't a lot of growth in this space, and it isn't to say that there aren't things coming that will. So I think what's commercially available, that when you go to the supermarket and try and just buy it, I'm not sure how much of an impact some of those have. But there are some products that have limited evidence base at helping in some different conditions. So there are some probiotics that have been shown to have effect in patients who have difficult IBS symptoms. And there's a sub group of kind of inflammatory bowel conditions. So in people with ulcerative colitis who have had to have an operation to remove their large intestine, and they've then had a restorative procedure done to form something called an ileoanal pouch. There was a type of probiotic called VSL3 that had evidence to help that it could help treat pouchitis, which is inflammation in that pouch that's formed. But what is troubling at the moment is that the form of VSL3, so the company that produced it, got taken over and they changed the formulation of the product but didn't repeat any of the studies. So it's still got the name VSL3, but it's not the same product that was proven to be effective in these studies. So I think there's lots more growth that will come in this area, and my hope is that we'll find that there are more and more effective treatments. But in terms of what's available out there at the moment for treating IBD, we don't have great evidence for them.
Thank you very much. Do you think fasting would do any good for colitis?
Uh, I wouldn't recommend fasting broadly. I think what you might find is that in people with difficult symptoms, they will often find that as soon as they eat, they need to go to the toilet. And so if they don't eat, they think, well, okay, then I'm getting less symptoms. And so, you can't dispute the fact that that might have short-term help with controlling your symptoms, but it certainly doesn't have positive impacts on your bowel inflammation. And in the longer term, obviously, repeated periods of fasting when you have a active inflammatory disease is going to be quite harmful to you in terms of losing weight, losing muscle mass. So we don't tend to recommend fasting, and we don't have any evidence that would say it's a helpful thing. But we appreciate that there are some circumstances when you have really severe symptoms and the second you eat anything, you need to rush to the toilet, why people might wish to do so.
Okay, thank you. This has been a fairly popular question. And forgive me again for my pronunciation if it's wrong, but is microscopic colitis considered worth treating as it's not as bad as the others? Or do you think people should just live with it? That was not my
No,
question. It's
no.
obviously somebody else.
So microscopic colitis is, again, it's another area of real kind of interest. So microscopic colitis, whilst it doesn't it can, I think what I'd say is it can be really disabling still. So people can still have very severe diarrhea. People can still find that they're having to rush to the toilet dramatically, and people are still finding that they're having to change their lives dramatically as a consequence of it. So we mustn't underestimate the impact that it can have on patients. There is interest in looking at FMT and microbiome intervention in microscopic colitis, although it hasn't, you're right, been given the same level of attention yet. And that's something that may come and may be kind of a natural follow on for the work that we're doing. If we can demonstrate that it's effective in inflammatory bowel diseases, then you'd have more justification for them making that intervention happen. I think microscopic colitis is one of those conditions that doesn't, it hasn't been given the same attention across all the different things. So in ulcerative colitis and Crohn's disease, obviously we have all these different drugs that come to market that come with a high cost and have their limits of kind of benefit. And none of the companies that make them are looking to do studies in microscopic colitis to show that they work. So a lot of the kind of organizations in gastroenterology that we all sort of are members of are looking at getting data from people that are involved in treating the condition to see, of course, looking at microbiome intervention, but also looking at whether any of these drugs that are now licensed in inflammatory bowel disease could be used to treat microscopic colitis more effectively as well. So it is something that I hope will have more options to treat. And certainly in cases where I've had people with very disabling symptoms that have been not responding to our usual treatments, I have considered using some of those drugs. And we've seen patients who have been very keen to have fecal microbiota transplantation, but just in the current sort of climate, we can't justify using it outside of a trial setting. And that's the challenge with FMT. We don't want to start using it outside of just as sort of for one-off intervention or indications unless we're doing it as part of a study that's going to allow us to more broadly start to do that in the future.
Okay, thank you. I think we've got time for one last question. And I'm sorry because there are more questions on the chat, but I'll make this brief. Do you think that FMT could have an indirect effect on pelvic floor function in IBD patients through its impact on the bowel inflammation?
Yes, it's a very specific question. So I think if you're better controlling inflammation, then that will inherently improve continence and control, because if you're passing more formed stool. So it depends on the type of bowel inflammation that they have. But if patients have complicated rectal inflammation, and you can reduce that with an intervention, and it may well help pelvic floor function to a degree. Can I say it's directly impacting upon pelvic floor muscles, for example? Not necessarily, but yeah, if you're reducing inflammation and consequently producing more formed bowel motions, then it will be easier to manage that from a continence perspective, and hopefully will make some of those really difficult symptoms easier to manage. So we would hope it could be part of a benefit, but I don't know whether, because we don't have the same level of evidence in Crohn's, which is where you'd get more complicated perianal problems as well, then it's so certain. But I think it could be part of a strategy that could help symptoms, certainly.
Okay, well, thank you very much. I'm sorry, I'm going to sneak in one more question if you don't mind, because
Sure.
this interest. Do you think there is any link between HPV, as you mentioned it, it can lead to the development of cancer?
Mhm. Is there any between HPV and the development of cancer?
Is there any, or it says, is there any link between HPV, as you mentioned, it can lead to the development of cancer?
Oh, is there any link between the micro? So IBD and HPV, or I guess so. HPV is human papillomavirus. So there are multiple different HPV viruses, and certainly they have been linked with cancers, specifically, well-established links to cervical cancer and to head and neck cancer. There's not been a lot of study of it in the context of inflammatory bowel disease, and broadly, even the virome, so all these different viruses that are present within our bowels, perhaps understudied at this point. And so I don't know that we can use microbiome intervention looking specifically at HPV, and the bacteria that I was talking about, it was Fusobacterium nucleatum, which has been associated with colorectal cancer and is one of these typical oral bacteria that is also seen in greater numbers in patients with inflammatory bowel disease and more severe inflammatory bowel disease. But we don't have the same information about HPV, but it's certainly, HPV does cause, or particular types of HPV cause certain types of cancer, yes.
Right, okay. Well, thank you very much. And I'm sorry, because I know there are some other questions out there, but that's been really insightful. Thank you very much to people who have asked those questions as well. And there were a lot of people posting some brilliant questions, and there's been some really lovely feedback as well for you, Dr. Rimmer, as well, to say thank you very much for this talk. But I think it's really important that you acknowledge people that have been part of the trials as well as the team. Without the members of the public taking part in those studies, and putting themselves through this as well, we don't learn more, do we? We don't get to do these studies and find these different developments. And what an amazing gift that is to do for people.
Yeah, and I think
Sorry.
I'm incredibly grateful to all of those patients that help us out so much to try and make these small changes to have a kind of long-term better treatment. So yeah, immensely grateful.
Yeah, so I think, again, if people want to take part in a trial or a study, it might be that they want to register, and we're going to be putting some links in the chat now as well, so that you could click onto those and have a look at that if you're not already aware of the Be Part of Research campaign, which the National Institute for Health and Care Research, thank you very much, Shilpa's just popped that in the chat for us. And again, just type that in if you've not already known about that, or tell other people about it. And you could do that. But I've got to say that from my perspective, I think it's so important that we do hear from people, isn't it, Peter? We want to get people involved in our research. We want to hear from them from the very start. And I know that's been very much part of what you've done. You might recognise some of the people on these pictures because they are part of the liver and GI group that we work closely with, for instance. And we've done lots of different events to try and engage with people, but also to involve them so that they are properly working with us from the very beginning. And again, we won prizes last year, again, for the work that we've been doing. We're delighted to get people to be part of it by signing up and hopefully, being part of perhaps groups that we run, or we'll put you in contact with different things that you can get doing via newsletters. You can choose a different way that you get involved with us, and we'd be delighted to hear from you. Again, we're going to put that in the chat we've just done. Thank you, Shilpa. And then finally, if you thought this was good, then we would love you to tell other people about it. It's recorded, you can go back and watch it, but also you can sign up for the next one, which is going to be next month by Professor Andrew Filer, who's representing the inflammatory arthritis theme and talking about the research that they've been doing. And he's very familiar with about understanding the joint properly. So it's inside the joint using ultrasound to understand and treat arthritis. So that's the 19th of June at 6 till 7 p.m. again. And again, you get the opportunity if you attend live to also be able to ask questions. And it will be recorded, but I would just like to say a massive thank you to everyone for joining the call tonight, and Dr. Peter Rimmer for giving us your time and expertise. So nice to see you all. Take care. Have a good evening. Goodbye.